This is the second half of Chapter 10, where we're going to focus on some specific examples of tumor suppressor genes. And a lot of these are genes that we saw in our Chapter 8 over cancer genetics. As if you remember, a lot of those inherited mutations were in various tumor suppressor genes. So you've seen a lot of these genes before, but I want to dive in and give you some more information on what these genes do, how they function at the molecular level, to give you some feel for why mutations in these genes are a specific problem. So we'll start with retinoblastoma, which we've seen quite a few times throughout the semester. You remember retinoblastoma functions at that first DNA checkpoint before you go into S phase of the cell cycle. And retinoblastoma functions by binding to this transcription factor called E2F. Um, if retinoblastoma is phosphorylated by CDK cyclins, it will release E2F, which will then go into the nucleus, activate transcription of genes that are necessary for uh, proceeding through S phase, right? We basically start making all the enzymes that we need for DNA replication. And also, just to take this back to the previous chapter, right? Remember, cyclin CDKs are commonly activated by that RAS pathway that we discussed. So I know we've seen that before, retinoblastoma. We've also talked about P53 quite a few times, but we didn't really get into how P53 does its job. So you might remember this image in the upper left corner of the screen here, where we talked about P53 being part of this internal apoptosis pathway where you get DNA damage, activating P53, eventually leading to the caspase cascade and apoptosis. Um, so let's dig into some of the details here. What exactly is P53 doing and how does it function in this capacity? So it turns out what happens is the DNA damage actually activates this protein called the ATM kinase. And ATM kinase would then phosphorylate P53, which would release it from its interaction with this protein called MDM2. MDM2 would normally, excuse me, normally target P53 for degradation via this process called ubiquitin-mediated degradation. And so let me just step away from this just to show you what ubiquitin-mediated degradation is. And this is a common mechanism for getting rid of unwanted proteins in the cell. And in a nutshell, what happens is the MDM2 um, is called a ubiquitin ligase. So it will actually add these ubiquitin molecules, little chains of ubiquitin, to the unwanted protein. And those ubiquitin chains are a signal to the cell that, hey, we need to get rid of this protein. It will then be basically dragged off to the proteasome, which is the digestive complex that will chew up the protein and just spit out amino acids and small protein fragments. Okay, so that would be the fate awaiting P53 if it were not for DNA damage and activation of ATM kinase. Right, so that saves it from certain doom. And once it's rescued, now P53 will go activate the transcription of two different genes, the P21 gene and what's called the PUMA gene. Now, P21 is a CDK inhibitor, so it's going to inhibit the activity of CDK cyclin, which is going to cause cell cycle arrest, right? Because remember, CDKs and cyclins are driving the cell past those various cell cycle checkpoints. So if we produce P21, we're going to go into cell cycle arrest, which makes sense, right? If we have damaged DNA up here, we don't want to go continue through the cell cycle. The other branch here, the PUMA protein that would be produced, is going to inhibit BCL2. And you may remember, let me go back just a couple of slides here. This is small, but we've seen BCL2 before. That is the protein, if you look in the upper left here, that sits on the surface of the mitochondria, basically protecting the mitochondria from these death-promoting proteins. So it's BCL2 is basically actively fighting against this apoptotic pathway. Okay. So if we inhibit that via PUMA, now we've removed this obstruction, so we're going to proceed to apoptosis even more quickly, right? And again, that would be a good thing. If we have DNA damage, we want apoptosis. We want to stop progression through the cell cycle. So if we mutate p53, then we're not going to produce those proteins, right? And we are going to continue through the cell cycle. We're not going to carry out apoptosis. And so I just wanted to point out that p53 is a tetramer. So in a normal cell, you have your two copies of the p53 gene that are both producing active p53 proteins, and we have to bring four of those proteins, four of those polypeptides together into this tetramer to have an active, biologically active functional p53. And so if we're thinking of p53 mutations, you could, of course, have, changes in the p53 nucleotide sequence. We would have to inactivate both copies potentially, right? So we're not making any functional p53 protein. That would obviously be a problem. You may be able to function as a heterozygote, right, where you've got one normal copy of the p53 gene, one mutated copy that's not making any p53 at all. And in this case, we could still form tetramers, right, because we're making that functional protein. But there's this interesting phenomenon, and this really applies to any protein that functions as a tetramer in this way. And that's this phenomenon of, excuse me, what's called dominant negative mutation. So we'd say p53 and similar proteins are susceptible to these kind of mutations. And this is kind of unique because what we see happening here is, in this case, we've got a cell that is heterozygous for a p53 mutation, but the mutation in the p53 gene is still allowing protein to be produced. It's just that protein is not quite functional, but it will still integrate into these tetramers. So we're basically getting half of our p53 protein is normal, half is this mutated non-functional protein that can still incorporate into tetramers. And so what will happen is pretty much all the tetramers that get made are going to look like this, right? Where half the subunits theoretically would be normal, the other half are mutated, and these two mutated copies are basically overriding the normal copies. So that's the dominant part here. And the negative part of dominant negative is now is having a negative effect on the function of p53, right? It's basically wiping out its function. So there are cases where even just a single mutation in one copy of the p53 could be enough to cause a cellular problem. And I think you may remember from the genetics chapter, Lee-Fraumeni syndrome, we talked about the fact that p53 seems to be particularly important in lots of different cell types, because if you have p53 mutations, your risk of lots of different types of cancer increases. But you can see from this graph, you know, there are some cell types where it does seem to be more important than others. So if you have p53 mutations, you're more likely to have cancers at the top of the list rather than the bottom. But in general, p53 is a bad one to mutate. You would like to keep your p53 as healthy as possible. All right, sorry to jump right into this. We've seen this picture before. You were probably hoping to never see it again. But the third example I wanted to give you was that APC gene, which you might remember was associated with this hereditary form of colon cancer. This followed that two-hit model, very similar to retinoblastoma, where you inherit one mutated copy of the APC gene, and then it's very likely throughout your life you're going to pick up a second hit in the second copy, and that's going to lead to colon cancer. And individuals with this inherited APC mutation, their colon is basically just covered with polyps, as you can see here. Um, and so the APC gene is part of this signal transduction pathway called the Wnt pathway, which is very critical during embryonic development, but as I'll get into, is also particularly important in the growth and maintenance of colon tissue. So let's get away from that picture. Okay, so what does APC do? Well, so as part of this Wnt signaling pathway, and it is a member of this what's called a destruction complex. It's a protein complex that will target this transcription factor called beta-catenin for ubiquitin-mediated degradation. So basically, this complex, APC, is trashing this transcription factor, beta-catenin. And normally, this would be the default state if you don't have, if the cell is not receiving any of this Wnt signal. Okay, so this would be sort of your baseline. And then if this Wnt signaling molecule shows up, and this again, this is in normal cells, what will happen is that's going to activate proteins that will now inhibit the destruction complex. So inhibit APC, that allows beta-catenin, the transcription factor, to go to the nucleus and start transcribing genes that are going to be necessary for cell division. So basically that Wnt signal is kicking on cell division by inhibiting APC or its associated complex. And so what we're seeing in individuals that have APC mutations is that destruction complex is not active, right? Whether or not you have Wnt signal, it doesn't matter, right? You have a cell like this where there is no Wnt signal, but because that destruction complex is no longer active, beta-catenin is just always free to go to the nucleus, and we're always going to get cell proliferation. And so just to put this back in the context of colon cancer specifically, as I mentioned, that APC gene and this Wnt signaling pathway are particularly important in terms of just maintaining the intestinal lumen in the colon, right? So it's the epithelial cells lining the surface of your colon. And you have these physiological structures that are called colonic crypts. They're basically just these deep folds or invaginations in that cell layer. So you have the crypt down here is kind of like the bottom of the fold. And what you see is down here in that crypt, you have a bunch of stem cells that are receiving Wnt signal actively from these nearby stromal cells. So that Wnt signaling molecule is binding to receptors on the surface of these stem cells. That is, as we just talked about on the previous slide, triggering cell division. As these cells divide, they get pushed up out of the crypt, and as they move away from the stromal cells and the source of the Wnt signal, they stop dividing and they start differentiating. But in the situation of like the APC mutation, even as those cells move out of the crypt and away from the stromal cells, because they don't have that destruction complex, right? They're going to continue dividing anyway, and you're going to end up with a polyp, which is what you see here. So it's literally just going to be polyp after polyp after polyp. And those polyps by themselves are not enough to be cancerous, but as we discussed in the genetics chapter, it's very likely one of these cells in the polyp is going to pick up a second APC mutation, and then that very likely is going to be enough to cause a true cancerous state. All right, fourth example is in this what's called the P10 or the PI3K pathway. And this is an interesting one because you might remember in the oncogenic chapter we talked about RAS and the RAS signaling pathway. And I mentioned that RAS was part of three different signaling pathways. We focus primarily here on the middle pathway with the MAP kinase cascade, etc. But we had this pathway on the left here that could lead to stimulation of cell division, stimulation of protein synthesis, which would be important for growth, inhibition of apoptosis. And so this is going to be an example of a tumor suppressor gene that's basically sitting in the middle of an otherwise oncogenic pathway, right? So we talked about RAS as being sort of the prototypical oncogene. So this is an oncogenic pathway, but we do have this tumor suppressor gene right in the middle. And I don't want you to get hung up on the details here of this particular pathway, but I wanted to draw your attention to this P10 protein here sitting in the middle of this pathway. And if we follow this pathway through, we're basically activating this protein kinase B, which is another oncogene. It would inhibit apoptosis, inhibit cell cycle arrest. And the P10 protein here in the middle of this pathway is basically fighting against this pathway. So it's converting this intermediate PIP3 back into PIP2. So it's actively working against the progression of this pathway. So putting the brakes on it essentially. So if we mutate P10, if we cut those brakes, then we're going to race through this pathway uninhibited. Okay, so you can have tumor suppressor genes even in the context of these oncogenic pathways. Next example, this one doesn't get talked about a lot, but I think it's really unique, so it's worth mentioning, is this CDKN2A tumor suppressor gene, which is particularly interesting because it actually produces two tumor suppressor proteins from a single gene. So I don't know if you're familiar with the phenomenon of alternate reading frames, but this would be a gene that actually has two different start codons, and depending on which start codon you start translation from, you produce two entirely different proteins. In this case, both of the proteins happen to be tumor suppressor genes. So the two proteins you can produce from this gene are P16 and ARF. P16 will inhibit CDK cyclin, so it's basically blocking progression through the cell cycle past those checkpoints. ARF is inhibiting MDM2, which you might remember was going to basically get rid of P53. So this would basically be an inhibitor of apoptosis. So, if you were to mutate this one tumor suppressor gene, you could potentially eliminate two tumor suppressor proteins, which would obviously not be ideal. All right, so just to remind you, we talked about the fact that tumor suppressor genes could be gatekeeper genes, meaning those that help control progression through the cell cycle, or they could be caretaker genes. Those would be tumor suppressor genes whose normal job is to take care of the DNA, basically fix DNA damage. And if you have mutations in caretaker genes, that typically leads to what's called genetic instability, meaning just a large increase in the number of mutations and DNA damage that can happen because you're no longer fixing it. And if you remember, we talked about the XP example. This was again from our genetics chapter, where the genes or part of this pathway were involved with repairing thymine dimers, for example. So the example I wanted to give you was the BRCA genes. We mentioned those previously a number of times, but these are of course famously associated with certain hereditary breast cancers and ovarian cancers. And we're saying this is totally rad because the BRCA1, BRCA2 genes are part of these rad protein complexes. So there's a protein called rad50, rad51. And even though we don't exactly understand what the BRCA proteins are doing as part of these complexes, what their specific roles are, we do understand what the complex in general does. So the rad50 complex that BRCA1 is a part of is called an exonuclease complex, and you may remember BRCA1 and BRCA2 are involved with repairing double-strand breaks. So what this rad50 complex with BRCA1 will do is chew back individual strands of DNA right around the break. So we're going to get these single-strand overhangs. Okay? And then that's going to open the door for the rad51 repair complex with BRCA2. This will come in, and the rad51 will coat those single-strand overhangs. And I don't want to get bogged down in the details here, but basically we're going to be able to use those overhangs to repair the double-strand break. Okay? So BRCA2 is critical for that process there in some way. So again, if we have these mutations in either BRCA1 and or BRCA2, right, we're not going to be able to effectively repair our double-strand breaks anymore because those complexes are not going to be fully active. And while we're on this idea of caretaker genes, I also wanted to mention the spindle checkpoint, because this is a very common phenomenon in cancer cells. Is you'll see lots of aneuploidy, right? Weird numbers of chromosomes. So here you see a karyotype on the left from a normal healthy human cell, right? We've got our 2 copies of chromosome 1, 2 copies of chromosome 2, etc. Everything looks great there. Here we have a colon cancer cell on the right, and you can see things are very, very wrong, right? We've got 3 copies of chromosome 1 and 3 copies of chromosome 2, and 4 copies of chromosome 11, so we've got lots of aneuploidy going on here. Extra chromosomes, missing chromosomes, lots of translocations. And so we think this aneuploidy, or really we know this aneuploidy is due to defects at the spindle checkpoint. So this is the checkpoint right in the middle of mitosis. And what's normally going on here at the spindle checkpoint would be the cell is trying to make sure that the mitotic spindle, the microtubules, are correctly attached to all the chromosomes. Remember, each chromosome is made of 2 sister chromatids. Those sister chromatids are glued together by this protein called cohesin. And normally, once the spindle is attached correctly, this enzyme called separase gets activated by what's called the anaphase promoting complex. That makes sense because we're getting ready to go into anaphase, separate the sister chromatids. So we activate separase, it chews up the cohesin protein, and now the sister chromatids pull apart and we go into anaphase, right? Well, the sort of the quality control here, the proteins that are making sure the spindle attaches correctly are what are called the MAD and BUB proteins over here. So they're kind of lurking around, waiting to make sure that the spindle attaches. And while they're lurking around, they will bind to this protein CDC20 that was an important part of that anaphase promoting complex. So until the spindle attaches, this anaphase promoting complex cannot be activated, right? But once the microtubules that are part of the spindle do come in and attach, the MAD bub proteins release CDC20, and then we're off to the races, right? We activate separase, etc. So imagine you have mutations in the MAD and bub proteins. Now this quality control is gone, right? And cells are going into anaphase even when they shouldn't, right? Even when the spindle is not attached properly. And so that's going to lead to all kinds of non-disjunction events and therefore lots of aneuploidy. All right, so just to wrap things up, we've talked about lots of oncogenes and tumor suppressor genes, and you may get the feeling that there's like a million of each. but our current estimates are that there may be over 100 different oncogenes and at least 2 to 3 dozen tumor suppressor genes that seem to be biologically active. So even though that is a lot, it is a finite number. And I also want to remind you that usually mutations in just one oncogene or one tumor suppressor gene are not going to be enough to cause cancer, right? Cancer is usually a multi-step process. That's the whole reason we don't usually develop cancer until later in life, because it takes time to acquire all these mutations. And this process has probably best been studied in colon cancer, or at least it was first really nicely illustrated there. And the Emperor of Maladies book that we're reading also kind of goes through a nice example at the end of this kind of progression. But we were just talking about colon cancer and the APC mutation, right? So that might be the first step towards colon cancer would be an APC mutation. That might give us the beginning of a polyp, let's say. But then, you're also going to have to mutate additional oncogenes and tumor suppressor genes. We've got a RAS mutation, a P53 mutation. So we've got to pile on mutation after mutation. And as we do so, we're just seeing this progression in the pathology, right? From normal to slightly abnormal to, very aggressively abnormal metastatic cancer. So really, if you time this out, you're potentially looking at 20 to 40 years, right? The amount of time it would take for all these mutations to accumulate in a single cell. All right, so just to finish here, kind of tie together the last few chapters that we've talked about. We've got our normal DNA that is constantly under attack by different mutagens, things that can cause mutations, radiation, chemicals, our unfortunate genetics in some instances, infectious agents, right, leading to gain of function mutations in oncogenes, loss of function mutations in tumor suppressor genes. As those things begin to accumulate, we can also throw in epigenetic changes, right, changes in methylation patterns. But collectively, all of these things are going to lead to some of those key hallmarks of cancer that we talked about, right? I won't go through them all again, um, but hopefully that's bringing together some of the varied ideas that we've had in the textbook up to this point. So, all right, thanks for listening, and we will pick this up in chapter 12. We've already covered 11, so we'll be on the last chapter of the book next.